Okay, so today we're gonna move up our pyramid. We said this pyramid, we're gonna be basing all of our anatomy and physiology on it. So we have different layers, and we have different structures, different anatomical structures, and different functions that we are going to be taking in order. So we're not going to be studying things all over the place. So we have a pyramid, and we said in this pyramid, we have the cell, that is the most basic level, the cell. Then many cells are going to give us tissues. So tissues are collections of the same cells. So same cells coming together, nerve cells coming together to give me tissue. Muscle cells coming together to give me muscular tissue. Bone cells coming together to give me bone tissue. Then when you move up to organ, now you will see that you have different tissues now coming together to give you an organ. You will see nerve tissue, blood tissue coming together with epithelial tissue to give you the skin. Then you have multiple organs connected together, and that is going to be the organ system. That's going to be the organ system. That's going to be the organ system. And then after the organ system, you can have the organism, and many organisms give us the population. Okay, below the cell, there is a more basic level known as subcellular level. That is where you have the micromolecules, the macromolecules, you have the organelles, you have the enzymes. So all of these guys, they make up my cell and many cells. So without them, I'm never going to have a cell. Without them, I will never have a cell. Without the cell, I'm never going to having a tissue. Without the multiple tissues, I will never have the organism. So this is the most important. The more down you go, the more basic, the most important. So you can never go to high school unless you had middle school. You can never go to middle school unless you had elementary school. You can never go to college unless you had high school. So the basic means the most important. So this is more important than the cell. What's happening inside the cell, and we proved that. You said, with cancer, you had something wrong with an enzyme. Okay, you have something wrong with an enzyme that is supposed to stop the cell from uncontrollable cell division, but the cell is now dividing uncontrollably, not because the cell, but because something inside the cell. You had an apartment, the apartment burned to dust because the microwave, something was wrong in the microwave. But people don't look at the stuff that is sub-apartment, that is below the apartment. But actually, they are the ones that make your apartment cannot stand on its own. You need these supplies there. So now we have four basic types of tissues. We have four basic types of tissues. We have the epithelial tissue, that is where you find your skin. That's the most important. The epithelial tissue. So we cannot go around not knowing what are our four basic tissues. The epithelial tissue, then you have two tissues that are very similar. But before that, you have your epithelial tissue, you have your something like your blood, your connective tissue, and then you have two that are very similar. And these are your nervous tissue and your muscular tissue. What is so similar between nerve tissue and muscular tissue? Both are excitable. Nerve and muscular tissue are excitable. Nerve and muscular tissue are both excitable. But epithelial tissue and connective tissue are not excitable. They have different roles, as we will see in a little bit. So one of them is excited by involuntary input. Very good. The other one, so the nervous system, it has two divisions. It has an autonomic nervous system, and then it has this autonomic nervous system, and then in the autonomic nervous system we have sympathetic and parasympathetic. We have voluntary and involuntary. We're going to get to that. But for now, both of those are really similar. That's how you're going to know the four. So you are responsible of knowing these four tissues: the nervous tissue and the muscular tissue. Those two are they come together. And then the other two are your connective tissue and your epithelial tissue. Okay, so the organ is a structure with discrete boundaries. Structure with discrete boundaries that are composed of two or more tissue types. So you see, the organ must have, the tissues are the same. Tissues are the same, but the organ must be multiple different tissues. So you cannot have an organ that is made up of the same tissue. There must be different tissue. Histology. In histology, you have microscopic anatomy. So our understanding of what the cell is was based on histological section. So if I have an egg that looks like that, and this egg, I don't slice it coronally, sagittally. I do not slice this egg frontally or sagittally or transversely. Will I ever know that there is a yellow yolk inside the cell? If you don't slice it in half, will you ever know? You will not. So that's why we devised these different histological sections. We said that if you slice something in half and you end up with right and left, you end up with right and left. You end up with right and left. That's an example of a sagittal cut. But you cut right and left, 50% to the left and 50% to the right. 50% to the left and 50% to the right. Okay, what if I cut also sagittally, but I end up with 80% on the left and 20% on the right? What kind of cut is that? If I cut, so let's say I do this. I have two structures like that. I cut both of them like this. So now I've cut them in half. I have two cadavers. I've cut them like that. So I have a right side, anatomical position, left side. And we have a right and a left that are both equal, 50% right, 50% left. But if I cut like that, and I have 20% right and 80% left. 20% right, 80% left. Just like that. So you cannot tell me a different name. If you give me a name for this, it's the same. The issue here is how did I slice them? So what is that when I cut them both in half? But in half, I get right and left, not front and back, because I can cut in half like this, end up with frontal and anterior, posterior, ventral, dorsal. But now I'm cutting in half. I'm having right and left. I'm having lateral and medial. What is that called when I cut right and left? Yeah, that's a sagittal. Excellent. That is a sagittal. Sagittal cut. What about this one? It has to be like this one. It's also sagittal. But this one that's right in the middle is known as mid sagittal, and this one is known as, that is mid sagittal, and this is known as parasagittal. That is parasagittal. So, and then oblique is like you're tilting the transverse. So transverse, you have up and down, superior, inferior, cephalic, caudal. That is transverse. But when you tilt if you tilt the transverse section about 30, 40, or 50%, then you end up with oblique. Yeah, very good. Okay, so we define the tissue. The tissue is a group of similar cells and cell products that are working together to perform a specific role in an organ. You gotta know your four primary tissues. What are the four primary tissues? We said two come together because they are excitable. What are they? Nervous and muscular. What are the other two? What are? Very good. So we're also going to be examining the relationship of these tissues to the extracellular matrix. You'll see some tissues, they have more extracellular matrix, and others, they compress more against the extracellular matrix. So this extracellular matrix is what is surrounding. So you don't see brick, even though you know this is a brick wall, but you don't just see bricks. There is cement. And on top of the cement, there is this paint. So the extracellular matrix is what's going to be painting on top of our cells. So you would have fibrous proteins and this clear gel known as the ground substance. That's like a watery extracellular matrix fluid known as the tissue fluid or the extracellular fluid. You see, the extracellular fluid has another name, and that is the interstitial fluid. Why? It is because interstitial fluid is found between the cells, and it gives like a support and cushion for the cells. And this interstitial fluid, this extracellular fluid, is rich in electrolytes, is rich in proteins, is rich in nutrients. So now it depends on the cell. Does the cell need more electrolytes? Does the cell need more sodium? Cell need more potassium? We know that the cell membrane is very dynamic. Your cell membrane at this moment will never look like my cell membrane at this moment, because you may have had a meal, I may have had a different meal, I may be starving, I may be a diabetic patient. So what's happening at the surface of my cell is never going to look the same as what's happening at the surface of your cells, on your tissues, even though they are the same tissues, but they have different functions. And there's water, gases, as we said, minerals, nutrients, waste, and hormones. So these tissues, these four tissues, they arise from different germ layers. So you know we begin with a one cell. What's the one cell that we begin our development from? The zygote. So you have, is the sperm a cell or not a cell? Hold on. Can you please say your answer again, Rahim? Is the what? Clump. Is the sperm a clump of cells? Okay, do you think the sperm is a cell? If you think it's a cell, raise your hand. If you think the sperm is not a cell, raise your hand. The sperm is a cell. It just has half of a DNA. So when it binds the egg, now they make. So the sperm, remember, it has a nucleus. It has a nucleus with one X, and the egg has a nucleus with a X Oh no, always X. The male is X or Y, but is one N haploid called haploid. It has one N, then they bind together. You get your two N. Then that's your zygote. That is your first two N, the zygote. This zygote, this zygote is going to undergo cleavage. It's going to undergo cleavage, and this cleavage is going to this cleavage is going to have the cell enter multiple rounds of cell division, and then you're going to have the growth, and then you would have the germ layers to begin to fold. Okay, you have to know the same way we knew what our four tissues are: the muscular, the nervous, the connective, the epithelial tissue. Now you gotta know where they arise from. So we have three different layers from which the germ layers arise. You have from the name, ecto is out, ectoderm. Endo is inside, endoderm. And in the middle is the middle mesoderm. So you have to know that. Ectoderm, very outside. In the middle, mesoderm. All the way down, that is endoderm. So now you knew what they were, the ectoderm, the endoderm, and the mesoderm. So now you got to know these tissues, they give rise to what? So the ectoderm, because it's on the outside, it makes perfect sense. It gives the skin, the epidermis. So if I ask you on the exam, which germ layer give you the skin, you know definitely that's the most outside, then that's definitely the ectoderm. Then you, as to the nervous system, then the nervous system is gonna, that's why your skin is highly rich in nerves. When you get an injection, you feel a pain when the needle hits your skin, but once it go inside, it don't hurt no more because your skin has more nerves than your muscle. So then makes perfect sense that the ectoderm gives rise to my nervous tissue. Then you have the endoderm. The endoderm, that is now the very inside, the core, the viscera, the very of the inside. What could be the most core? Obviously, remember in your digestive system, you eat something that does not get absorbed, like the people who drink milk and they have lactose intolerance. So they eat it this way and it gets out the other way. So technically the milk never entered your body, even though it entered your mucosa, but it did not make its way to the blood. And if it didn't make its way to the blood, it did not make its way to the tissue. That's why that xenobiotics video, you saw that all the xenobiotics or the pollutants or the medications, they trying to get to the circulatory system. Through your skin, they seep through your skin, they go through your skin to make their access to the blood vessels. You know, that's why the women with breast cancer, they put the castor oil poultice in the bra and they leave it there for a long time and it cured breast cancer because it seep right through the skin tissue. Yeah, that's very true.
They said you can put it in your navel also, and it helps with any GI issue.
Yeah, that's very true. And if you had a surgery, you had a scar tissue, you put the castor oil. So that all signifies what? That your skin is very quick access to the bloodstream. And also, who else gives another access, quick and valid access to the besides the skin? Yeah. The eyes. Was that in the video? The lungs, the respiratory system. Lungs is only the lungs. What about the nasal cavity, the oral cavity, and the trachea and the alveoli? So all your respiratory system, because you have a whole pulmonary circulation. Then what else do you have? The GI. So this GI has a mucosa. You know when you have a rhinitis, you are sneezing, you have a runny nose, you have sinusitis. It is the same. Rhinitis and sinusitis is the same. It's happening in different places. The mucosa layer of your nares and your vestibules, when they get irritated, that is called rhinitis. Then when you move down to the sinuses, the maxillary sinus, the frontal sinus, these also have a paste. They're pasted with mucosa. When the mucosa gets irritated, that's sinusitis. But it's the same. It's the inflammation of the sinuses. So here you will see that that is your endoderm, this mucosa layer. Nothing more below the mucosa, because the mucosa is touching your is the acid in the stomach. The mucosa is touching the acid, is interacting with the acid in your stomach. So that's the endoderm. So you can tell the mucous membrane lining the digestive system. There's nothing more basic to that. If you look at that's the stomach, and this is the small intestine, and then the large intestine. You see the mucosa is this layer. The mucosa is this layer here. This layer. There is nothing after it, just acid now. So that's the most in. That's why it's made in the endoderm. Then you have the middle layer. So now you know your ectoderm outside, your endoderm inside, and then you know your mesoderm. That's the middle layer. That is your gelatinous tissue, known as the mesenchyme. So I have to ask you a question. Where do we find the mesenchyme? Is that in the ectoderm, the endoderm, or the mesoderm? Yeah, mesoderm. So that an example of that is your bone. So your bone is right in the middle. It's not in the mucosa. It's not outside. That's why when the bone fracture and the bone breaks, it cuts the nerves, it cuts the blood vessel, cut the skin, come out the skin. It's called an open fracture. It's no longer in the middle. It now is on the outside. So your bone is not endo. Your bone is not ecto. Your bone is meso. Very good. Mesoderm. Okay, understanding histology requires awareness of how the specimens are prepared. So remember we said in the lab that we need to have a histological dye. So remember we manipulate the working distance, we elevate the stage, we lower the stage, and then we apply a particular histological dye to enable us to see better. So you use eosinophils, if you don't apply them with acid, you cannot see them, even if you have the right magnification. The basophils, if you don't apply basic blue, basophils, blue, basic. So the basophils are staining blue and they are basic. If you don't have the right staining, histological, physiological dye, you're never going to be able to see, even if the objective lens is working, even if the working distance is fine, you need to have the proper dye. So we use sections, and now let's examine these different types of sections. Tissue sliced into thin sections that are one or more cells thick. So there is a fixative section. That is the one they use in the cadaver because it prevents putrefying. Back in the day, they used to study the cadaver while the cadaver is putrefied because they didn't have this fixative staining. Now, when they apply this formalin, they will have better access to study it because it will not putrefy. It will prevent it from putrefaction. The stains also color the tissue, and they bind to different cellular components. So how did we know that the basophils stain blue and the eosinophils stain red? Is because the dye was able to bind to the granules inside the eosinophils and inside the basophils. The sectioning reduces a three-dimensional structure to two-dimensional slice. So when I do a cross section, I no longer look at the entire body. It's like when you do a CT scan. When you do a CT scan, do you look at the entire body, or you only look at two dimensions? So you're only examining length and width. You're not looking at the whole body. So you're just examining superficially. So remember that the sectioning is going to reduce my multi-dimension, my three-dimensional structure into two-dimensional. So I can have a longitudinal section. This is the coronal section, where you have a cut on the long axis, or you can have a transverse section. So you can have an even superior to inferior section, or you can have an oblique section where the tissue is cut at a transverse angle, angle between the cross and the longitudinal sections. So that's what we see here. You see, these are, if you cut in the middle, you will see like that. It's the same egg. If you cut in red, you will see this. This is two dimension. But if you cut in green, you will obviously see that 80% is on the right and 20% is on the left. So it will look like that, even though it's the same egg. You see, this is the same egg. When you cut it with blue, you just cut this little side out, and you're now examining it. You cut in red, you're looking at it in a two dimensions in half. And when you cut in blue, you're looking at just this really tiny um, um, section. So the most important thing you need to know is sectioning is going to reduce a three-dimensional structure into how many dimensions? Two. Very good. Now, if you look at this elbow, what is it called? Elbow mac and cheese? Elbow macaroni. Okay. Now, these are all, obviously, all sections. Now, look at blue. If you look at blue, you're just going to see that. If you cut blue, you're going to see this only. If you cut green, you're going to see that. And if you cut red, you will only see these two tubes. So the way we cut, we're going to break down the three-dimensional interpretation into two-dimensional images. Now let's look at how we examine our body. These are the three planes of the section. There is the longitudinal section. Now I was able to look inside the bone. I can see the epiphysis, the metaphysis. I never knew that the white bone inside it, there is blood. Who would have thought, had we not cut and longitudinal cut? Had we not cut longitudinally, how would we have ever known that the bone is where you make blood? We used to think that bone is important only for walking. Bone is important as a calcium salt or a phosphate salt reserve. If your body has some homeostasis imbalance with the calcium salt or phosphate salt, we go to our bone to degrade the bone. But now when we sliced it open, we see there is blood, because that is where we make the red blood cells. Also, we found that inside the core of our bone, there's fat tissue called yellow marrow. So that is known as the red marrow. And there's also the yellow marrow. There is a lot of fat, a lot of butter inside the bone, because your bone is growing and getting stronger, so it needs ATP supply. If you look at here, if you look at this blood vessel, if we cut it like that longitudinally, we're going to be examining. We are now able to examine the lumen of the blood vessel from the inside. But if we don't cut it transversely, we're now not going to be able to see the ratio. How much on the outside do you have bone compared to the core? And here we perform a oblique This is an example of an oblique section that enables us to see how these cells are structured one on top of the other. So now when you're cutting halfway, you will see how are the cells lined up on top of one another. So we need the more different histological sections we have, the more knowledge, the more access we are going to get to learn about the section. Okay, so interpreting tissue sections. So not all tissues are going to be sliced or sectioned. There is a smear and a spread. What's the difference between a tissue smear and a tissue spread? A tissue smear, the tissue is rubbed or spread across a slide. So we rub or we spread the tissue across the slide like a blood smear to see if there's a malaria, to see if there's some form of microbial infection that's going to happen inside the microscope. So blood and soft tissue like the spinal cord. Spread. The tissues are laid out on a slide. The tissues are laid out on the slide. So we place the tissue, we lay them out like the cobwebby tissues. These membranes and the cobwebby tissues, like the areolar tissue, you place it as is, and you don't smear it, you don't rub it, you place it holistically, and you examine it in a spread. Epithelial. Now let's begin with our first type of tissue. So we're going to do all of that to our first type of tissue, sectioning it, smearing it, spreading it, and we're going to be looking at the epithelial tissue in cross section, in oblique section, and longitudinal sections. So now, if you examine, this is the epithelial tissue, where there is a sheet of closely adhering cells, one or more cells thick. So with upper surface exposed to the environment or internal space. That's why epithelial tissue is found on the outside, and it is the core of the gland. So there's a gland that is full of some hormone, full of some enzyme, full of some sweat. Who's going to be the bottom? The core of that gland is going to be epithelial tissue. So how do we define epithelial tissue? Is a sheet of closely adhering cells. So they're like sitting in an auditorium. They're not sitting like you guys, one next to the other. One or more cells thick with upper surface exposed to the environment or the internal space. So that's one of the requirements. So the law to tell that epithelial tissue, one of its side has to face either the outside or the inside. Has to face the outside or the inside.
Do you have a picture of it?
Yeah, we will see it in a little bit. It covers the body surfaces. That makes a lot of sense. The body surface, the outside. How many surfaces we have? The outside, do we only have outside surface? What about inside? That's also a surface on the inside where the mucosa, where there's acid in the stomach, where there is the air going in. Remember we said you drink milk, you have lactose intolerance, or you don't have vitamin D to absorb it, it's going to flow right out. So now that's an example of you are hollow from outside and you're hollow from inside. And who's facing the outside or inside is always the epithelial tissue. So which of the four tissues is always facing the outside or the inside is definitely not the connective tissue, not the muscular tissue, not the nervous tissue. It's definitely the epithelial tissue. Very good. So that constitutes most glands, and the epithelial tissues are avascular. What does that mean? Avascular means, so how do they survive? if they have no blood vessels. You know they are tissue, so they are made up of cells. So cells, obviously, a cell is a cell, whether it's epithelial tissue cell, muscular tissue cell, nerve tissue cell, connective tissue cell, it's a cell. So the cell, in order to survive, need oxygen for oxidative phosphorylation, need oxygen for aerobic respiration. So now this cell, if does not get blood, how does it survive? They have blood, but not directly to them. They get an indirect flow of blood through the connective tissue layer right below them. So from they go to the basement. When they want to eat, they go to the basement. And that's how but they don't have it. So if you cut epithelial tissue, so have you ever had an abrasion but no blood? You never leak blood, and except you go down. So whenever you're bleeding, you know that you actually have went down to the connective tissue because blood vessels are flowing under the connective tissue. So the functions of the epithelial tissue. So why do we have epithelial tissue? The first function is for protection. The epithelial tissue protects the deeper tissues from injury. So your skin protects your muscle from injury. Your skin protects your bone from injury. From the inside, hollow, from the inside, your stomach. There's mucosa to protect your stomach from what? From acid. Because your stomach has a lot of acid to burn and to break down the food. So how is it that the acid in your stomach breaking down steak, but it doesn't break down the stomach itself? Because of the lining, the mucosa lining. It's like a snake has a lot of poison, but it doesn't kill itself. So, I'm sorry? The sphincter? Yeah. So if you are facing outside or you are facing inside, say no more. That's epithelial tissue. So they are there for protection. They are there for secretion. Remember I said that. If there is a gland, like a sweat gland or some gland like the pancreas secreting some hormone, mucus gland, enzymes, hormones, and other substances, they secrete a particular type of substance. So epithelial tissues are important for protection, secretion, also excretion. So the epithelial tissue is important for voiding and removing and excreting the waste from the tissues. Absorption. Epithelial tissues absorb chemicals. So remember, since they deal with the outside, they either secrete or they can absorb. So they can move in either way. So they absorb the castor oil from the skin and they secrete the sweat outside the skin. So they secrete mucus and they absorb gas, they absorb electrolytes into their core. So they are there for absorption and excretion. Also filtration. All substances leaving the body are selectively filtered by the epithelium. The epithelial tissue is avascular, doesn't have a lot of blood vessels, but epithelial tissue has a lot of nerves. A lot of nerves. That's why the baby, when the mom touched the baby, pat on the baby's back, the baby sleeps because there's a lot of nerves on the back. There's a lot of nerves for in the lips. A lot of nerves in the tongue. That's what helps with the taste, and a lot of nerves in the skin. So that's very important. That this imagine your skin, imagine your lips were made from muscular tissue. Imagine your lips were made, you're not going to be able to taste anything, and that kissing stuff not going to work because the nerves are highly vascularized in the lip area, and the um, the skin, tactile for the touching. But the muscle tissue, remember, when you get an injection, where do you only feel the pain? When you get a needle injection, you only feel the pain where? Yeah, in the skin. Once you go inside from the epithelial to the muscle, it's not going to be painful. So the epithelial tissue, so the general structure of the epithelia is cells are close together. We said they are like sitting in the auditorium. So there's a small amount of extracellular material. Epithelial tissue, as we mentioned, is avascular. There are no blood vessels in the epithelial tissue, and they are nourished by the underlying connective tissue. Do you see how our epithelial tissue is able to get its supply of blood? It's from the underlying basement layer connective tissue. Connective tissue. Who's highly vascularized with a lot of blood vessels? It's the connective tissue. From the name, connective, because it connects you to the blood vessels. So it's really highly vascularized. The epithelial tissue, they rest on a basement membrane. They rest on a basement membrane, which is a layer between an epithelium and the underlying connective tissue. So the basement membrane is the layer on which your epithelia are placed. The basement, please remember that. The basement membrane is the layer on which you have all of the epithelial cells lined up. Below the basement membrane, what do you have? Excellent, connective tissue. So basement membrane is basically not the line between epithelial and what? Connective, excellent. You said it. And you have, in the underlying connective tissue, you have collagen, glycoproteins, and other protein carbohydrate complexes. They anchor the epithelium to the connective tissue below it. Now, if we look at the general structure of the epithelia, so the surface, if you look at the surface of the epithelial cells, there's a basal, apical, and lateral surface. Let's begin with the basal surface. The basal surface of the epithelial cells is the surface that is facing the basement membrane. From the name, basement. They couldn't call it the basement surface. They just called it the basal. The basal. It's called the basal surface. So if you are facing downward, that makes you the basal surface. The apical surface is the surface that faces away from the basement membrane. The apical surface is the surface that faces away from the basement membrane. Is that clear? Okay. Then we have the lateral surface. The lateral surface is the surface between the basal and the apical surface. So what is the lateral surface? That is the surface between the basal and the apical. That's the middle surface. That is the side wall, the one that is in between. So the epithelia are classified by the number of the cell layers. There's simple epithelia. They have each cell anchored to the basement membrane. They have each cell anchored to the basement membrane. There's a stratified epithelia. Stratified epithelia have cells on top of one another. They have cells on top of each other. So they do not contact with the basement membrane. So that's the difference between simple and stratified epithelia. The simple epithelia have each cell anchored to the basement membrane. Each cell has access to the basement membrane. Like you guys, Every single one of you's foot is touching the basement, touching the ground. That is simple. But stratified, imagine you have another class of students. You are sitting, but some of them are sitting on top of your head. So not everybody has access to the ground. You are touching the ground, but their feet are touching your back. That's stratified. So who do you think is thicker? The stratified or the simple? Stratified. Stratified is really thick because you have two of them on top. So you have, yeah, so they're on top of each other. You see the stratified epithelium is what happens to the smoker. So when the smokers smoke the nicotine, smoking the weed, all of that is going to change the cells that are in the esophagus because there's so much stress. So they're supposed to be columnar simple. But because they're dealing with so much nicotine, they now have to undergo metaplasia. They got to change to keep up with the stress. Otherwise, they're going to get burned. So they convert, they become stratified. Then, if you keep smoking some more, they're going to become cancer cells because they're going to keep changing to keep up with this damage. So it's pretty simple. Simple versus stratified epithelium. Simple touching the basement, stratified on top of each other. There we go. How nice is that? Look at simple. Everyone is touching the basement. Everyone is touching the basement. Now look at stratified. Some are touching the basement, other ones are on your head. Now this middle layer is very tricky. This is a pseudo. Pseudo means halfway. Pseudo stratified columnar. So it's 2 in 1. 2 in 1. Each cell has some part of it on the basement and the other part on the head of some other cell. You get it? So here you're either you have your feet on top of the other cells or you have your feet on the basement. But pseudo stratified is 2 in 1. It's two at the same time. Each cell one time has a base, one has a foot on the basement, another one has a foot on the yeah, very good. This is squamous. So now this is a definition of the classes of the epithelium. You have simple epithelium, pseudostratified, columnar, and stratified epithelium. But still, this is a description of how are they aligned. When we describe the class of the epithelium, we're describing how are they aligned. Are they all aligned on the basement membrane? Are they all aligned on top of one another? Is each cell a little bit of this, a little bit of that, touching the basement membrane, touching the other cells? But that's just describing how are the cells placed. But now you got to look now at each cell. So now you can have many students' foot on the ground. But you're not describing me what kind of student. Is it a male student? Is it a female student? Is it a malnutrition student? Is it an obese student? Is it a middleweight student? Now the cell shape is going to tell you. Squamous, they look like an egg sunny side up. That's a squamous cell. Now you can take that squamous cell and make simple columnar out of that. So all of the squamous cells sunny side up, they're going to be touching the basement membrane. So we call that squamous simple. Simple squamous. If you take the squamous sunny side up and you put some on the basement but others on top of each other, now what do we call that? Stratified squamous. Very good. Very good. Then if you have squamous with pseudostratified, we call that pseudostratified squamous. So don't get fooled that we only have one way of looking at the cell. We look at how are they arranged, then we look at what they are. Are they sunny side up? Are they cuboidal? Cuboidal means the width and the length are the same. But columnar means you are more tall than you are wide. So you can have columnar cells being simple. You can have columnar cell being stratified columnar. You can have stratified simple. You can have cuboidal simple. You can have cuboidal stratified. So your body has different tissues based on the need. Based on the need, we have different types of tissue. Does that make sense? Yes, please. Pseudo, yeah, you're right. Pseudo means false, but sometimes pseudo means like you're halfway, or maybe like you're not, you're lacking a definition. You're like pseudo, so it's not, but you're definitely right. Pseudo means false. It gives the illusion. It gives the illusion. So this is pseudo stratified. So they're not actually stratified because each cell that is on top, it has one leg on top of another cell, but the other one is still on the basement. So it's not stratified, but it's kind of stratified, pseudo stratified. Okay? So now you see, now we're no more talking about the cell. We're no more talking about the nucleus, the cytoplasm, the rubber duckies, all of the we're not talking about that. We're done with the cell. Now we move up to the tissue. Then the next lecture, now we're gonna do the organs. Then after that is gonna be systems till you're done with ANP. Till you do ANP2, the male and the female reproductive system. So if you have some ideas, you're not clear to you, you gotta go back to the book, ensure you don't move forward unless you have covered all of the cell, all of the tissues, so that everything will make sense. Yeah. So now let's begin with the simple epithelium. That's right here. That's the simple epithelium. So when you say simple, you know everybody's touching the basement membrane. So there are three types of simple epithelium. These guys, you can have squamous simple, cuboidal simple, and columnar simple. You see? Simple squamous, simple cuboidal, and simple columnar. In the fourth type, you have pseudo-stratified columnar. You have pseudo-stratified columnar. So not all the cells reach the surface, but all reach the basement. They all reach the basement membrane. They falsely appear stratified. You see, you said it excellently. They falsely appear stratified. And some cells are taller than the other. Then you have these cells are important, these goblet cells. Goblet cells. They are goblet because they secrete mucus and saliva, goblet cells. They are like wine glass shape. They look like wine glass shape and they secrete mucus. They are simple columnar and pseudo-stratified epithelium. So now these false cells, so that's a high yield question. What's an example of a pseudo-stratified columnar? It is those goblet cells that secrete what? Mucus. So that here shows you, we're going to look at this in the microscope. You see the simple squamous epithelium, and these are the squamous epithelial cells, that's the alveoli, and these are the blood vessels. You have, this is a single row of thin cells. They permit a rapid diffusion of or a transport of substances, and they secrete serous fluid. The locations are the alveoli, the glomeruli, the endothelium, and the serosa. Now we're done with simple squamous epithelium. Now when you look at simple cuboidal epithelium, so now you have a picture, still simple. So everybody's touching the basement membrane. But the cells are not sunny side up. They are not columnar. They are cuboidal. It's a single layer of square or round cells. They are there for absorption and secretion and mucus production. And you find them on the liver, the thyroid, I'm not gonna ask you that. I'm not gonna ask you. Give me an example of where you find simple cuboidal epithelium, but you should know what it means. Simple cuboidal epithelium. You say, oh, simple means everybody's touching the basement membrane. Cuboidal meaning all the cells are as equally wide as they are long. So that's all you need to know. And the one example you need to know is the goblet cell. The goblet cell is the cell that does what? Pseudo-stratified, yeah. You see? They're wine glass shaped mucus secreting cells in simple columnar and pseudo-stratified epithelium. They come in two forms, simple columnar and pseudo-stratified. There's simple columnar epithelium. You see the brush border? There is a connective tissue. There is basement membrane. You see what is that an example of, guys? Is this simple or stratified? Can you please look at the way the mouse is pointing? What do you think is what kind of tissue is that? That's stratified. Every cell is touching the basement. That's simple. Now, simple is not enough. Is it simple squamous? Is this sunny side up? Is it? Yeah, that's simple column. You see column like a long, more than it is wide. Very good. So that's a simple columnar. And if you look at this one, they're all touching the basement, right? Can you see where my mouse is? But are they columnar?
No.
Are they sunny side up? They are what? So that's simple cuboidal. That's simple cuboidal. That is simple cuboidal. That's like we're looking in the even in the microscope, you're not gonna see it as good as this. But you still need to do the microscope to get comfortable with the lenses. Now, when you look at this, that's simple squamous. So now they are sunny side up. So these guys, you see the sunny side up egg? They are all simple. So they all touch in the basement membrane. You see? But they are sunny side up. You see the egg? Sunny side up. Squamous. We said squamous is sunny side up egg. So you see here? Squamous. Sunny side up egg. That's simple. They all touch in the basement. Simple squamous. This one, they all touch in the basement. Simple cuboidal. Very good. All touch in the basement. Simple columnar. Very good. What kind of cell is that? That the wine glass one. That's the goblet cell. You see your goblet cell now. See, and that's the actual. I'm sorry. Yeah, it can be the two. It is mostly pseudo-stratified, but can also be simple. Very good. Now what is this? Now if you look at it simply, you can tell it without reading. Is everybody here touching the basement membrane? Not these guys, so that's definitely not simple. What is that? Yeah, pseudo-stratified. Because you see this one, it has a foot on the basement and the rest is on the head of the other one. So that's pseudo-stratified and they are long. So they're pseudo-stratified columnar. Now we're done with simple. We gotta now move on to stratified. So in stratified epithelia range from 2 to 20 or more layers of the cells. 2 or 20 of the layers of the cells. So it's not like you have one somebody on your head. You got 20 on top of your head. That's stratified. That's the one we needed in places where there's like a lot of tension and a lot of stress. So we have this stratified. These stratified epithelia are named for their shapes, for the shapes of their surface cells. You have stratified squamous, stratified cuboidal, stratified columnar. Pretty simple. You have columnar cells that are on top of each other. You have squamous cells, sunny side up, on top of each other. You have cuboidal on top of each other. Now this one is important though. This urothelium one. That one you gotta know that one. That one is a question on the exam. I actually put it on the exam. This urothelium. A fourth type, the urothelium, is unique to the urinary tract. So this urothelium is an example of a stratified epithelium, but it is unique in the sense that it is really not stratified squamous. It is not stratified cuboidal. It is stratified is not stratified columnar. It is something that they call transitional epithelium. And it makes sense because the urine is going to burn. Urine is acid, is burning, is going to cut the bladder. That's why the bladder needs to be pasted with stratified epithelium. It makes no sense. It makes no sense that the bladder dealing with all the urine, this burning urine, is made of columnar. Does it make any sense that the bladder is made of columnar tissue? It has to be stratified. Tissue. So transitional epithelium. So that's just the urothelium. It has now its own name. Now they don't even call it the stratified squamous of the bladder. They gave it the name urothelium. So you know that's in the bladder, dealing with the urine. Stratified, dealing with urine. Stratified epithelia. So the stratified columnar is rare and is only in places where two other epithelial types are gonna meet. So stratified columnar is rare only in places where two other epithelial types meet. So the stratified squamous is the most widespread. It is the deepest cells are cuboidal to the columnar, and they include mitotically active stem cells. These stem cells are the progenitor cells that give rise to the other cells. So the daughter cells push toward the surface and become flatter as they migrate upward, and they finally die and flake off. You got to know this exfoliation. What is exfoliation? Disquamation. Exfoliation and disquamation is when they die. When they reach the surface, you know the stratified squamous, they are the deepest, and they start to rise as they mature. They go to the surface until they flake off and die and exfoliate. So they are dying and flaking off and dying. Hmm?
This has to do with the fibroblast.
So the fibroblast is a different type of cell. The fibroblast is not an epithelial cell. The fibroblast is there for healing with fibrosis to make scar tissue formation. Okay, there are two kinds of stratified squamous epithelium. There are two kinds of stratified squamous epithelium. There is keratinized or cornified, and then there is the non-keratinized. Keratinized is found on the skin. That's why they say keratin. And they are abrasion resistant. So you got this keratin protein that is going to resist all the abrasion. There are non-keratinized. So the non-keratinized, they are found lining the internal spaces, and they lack the surface layer of the dead cells. They lack the surface layer of the dead cells. So the keratinized stratified squamous epithelium. There are multiple cell layers, and the cells become flat and scaly. You see, this is keratinized. So we have stratified squamous, but it has a lot of keratin protein. What did we say keratin protein? You know, every protein cannot exist except in a dimensional form. It's either primary, no protein is working as primary. That's the transcript. There is secondary, tertiary, and quaternary. Keratin and collagen, are they secondary, tertiary, or quaternary? Keratin and collagen, secondary. Excellent. So you know some jobs, all you need is high school, and then you can do the job. You don't need anything else. Some job you need to go to school for. Some job you need to have the masters for. So some proteins secondary and they're done, like collagen. Collagen, to do its job, it should only be secondary structure. Collagen and keratin. Now, who must be give me an example of a tertiary structure protein? Tertiary structure protein. Hmm? I cannot hear. The proteins? Yeah, so they curve once, they go from primary to secondary. That's collagen and keratin. Now we go from secondary into tertiary. What's an example of a protein that don't need to be quaternary? Tertiary, it can get higher, do its job. What is that? Myoglobin. That's the oxygen storing protein in the muscle. Myoglobin. Okay, what's the protein that's you guys never heard of myoglobin? Myoglobin, myo muscle, myoglobin. So that's like the hemoglobin, but for the muscle. It provide oxygen. Okay, and we have the hemoglobin. Now that one is a quaternary. That's got to be 4. 2 beta, 2 alpha. 2 beta, 2 alpha. If one of the 4 structures is damaged, sickle cell anemia. So hemoglobin has 2 alpha, 2 beta. 2 alpha, 2 beta. When one of the beta has a mutation, they needed to have the amino acid glutamic acid, but now they have valine that becomes sickle cell now. So that's the quaternary structure. So keratin is the secondary. Now this is non-keratinized stratified squamous epithelium. They are the same as keratinized epithelium without surface layer of dead cells. They resist the abrasion and penetration of the pathogens. The location is the tongue, the oral mucosa, the esophagus, and the vagina. That is stratified. So you see all these areas are areas of stress. When the baby is coming out, imagine that vagina was not made from stratified epithelium, it was made from columnar. So the baby is going to do trauma to the mom. Or during the sexual intercourse, it's going to cause trauma. So that area must be stratified. The esophagus, you're swallowing, and you're drinking, and somebody drinking the alcohol, the aldehyde, the reactive oxidative stress. So imagine that wasn't stratified. It was simple. All that corrosive alcohol is going to damage the esophagus. The oral mucosa, always eating and always, crushing stuff. The tongue. So now you can have a sense that we need stratified where there is a lot of trauma to put up with, a lot of force. This is the stratified cuboidal epithelium. So you can tell they are cuboidal and they are stratified. This is the urothelium. This is the special one when we have the urine. This is the type of cell dealing with the urine. Multi-layered epithelium with surface cells that change from round to flat when they are stretched. They allow for the filling of the urinary tract, and their location is the ureter and the bladder. The ureter and the bladder.
The bladder stretches.
Yes, it has the ability to stretch and the ability to deal with the corrosive, this burning urine. It allows for the filling. and the distension. And it's full of many vessels, full of many nerve. So when your bladder is full of urine, you feel that, oh, I need to use the bathroom. So this stretch is going to distend the nerves. Okay? Yeah, that was a good observation. So, you know, the urinary tract is not just in the bladder, but since the urine is just coming out, so now that is acidic burning urine. So all of the path from the ureter going down to the bladder, then the urethra, that has to be pasted the same way your stomach is pasted with mucosa to put up with acid. All of the urinary system has this special type of really strong stratified epithelium known as the urothelium. Urothelium is multi-layered epithelium with surface cells that change from round to flat when stretched. So they have this capacity. When you stretch them, they no longer cuboidal. They become flat. They become like squamous. So they have the ability to modify. They can undergo conformation change to modify this urine. So that helps them to allow for the urine, for accommodating the volume of urine. You sound confused. So now the anus is not urine, that's not acid. So now that is going to be your large intestine. So that is going to be a tissue that is stratified also, but not like urothelium. It's not going to change its shape and dealing with urine because the yeah, it's obviously going to be stratified. Cannot be dealing with urine. Yeah, yeah. You know the sweat glands also stretch. No, that's not true because some you need to stretch to secrete sweat, to secrete a hormone like a gland that's soft. But we're talking about traumatized tissue, tissue that deals with urine, corrosive, injurious urine, or fecal matter, okay? Constipation. Now, that can cause fissuring, ulceration. So that needs to be stratified. So now we are done with the epithelium. Now we got to move on to the second type of tissue. You know, you see how they left the two together, the ones that are similar. You will see when we do one, the other one is going to be very similar. The nervous and the muscular, they come together. You're going to learn that if it wasn't for the nervous system, you cannot stretch the muscle. If it wasn't for the muscle, you cannot move the bone. Okay, so now let's do the connective tissue. Connective tissue is the most abundant, widely distributed, and variable type of tissue in which the cells occupy less space than the matrix. What is the most abundant of all the tissues? Muscular, nervous, connective, or epithelial? Connective tissue. Very good. Most cells are not in direct contact with each other. Most of the cells are not in direct contact with each other. They are, they have highly variable vascularity. They have highly variable vascularity. They are not in direct contact with each other, and some are highly vascular and some are not. Loose connective tissues have many blood vessels, and cartilage has no blood vessels. We will see that. Functions of the connective tissues. So the binding of the organ. So you know the skin is an organ. We said that the connective tissue is bound to the skin. So they connect one bone to another. This ligament, the tendon, that's connective tissue. The ligament connect bone to bone. So the muscle, they muscles to bone, skin to muscle, and hold organs in place. Okay, your liver is not gonna fall off your abdomen, and your heart is you're not gonna wake up and find your heart falling in your abdomen. Is staying stapled in place with this connective tissue. Support. They support the body and its organs, and they form internal framework of the organs. They form internal framework of the organs. They are there for physical protection, cushion. They cushion the delicate organs, and for immune protection, the connective tissue cells attack the foreign invaders. They attack the foreign invaders. Also, the connective tissue is important for movement. So bones provide lever system for body movement, and cartilages are involved in speech. Storage. They maintain stores of fat, calcium, and phosphorus, and it's also important for heat production. Heat production. So metabolism of brown fat generates the heat. So brown fat is a type of connective tissue. And for transport, the blood transports the gases, nutrients, waste, hormones, and blood cells. So we have fibrous connective tissue. That's what you mentioned, the fibroblasts. These secrete TGF beta, and the TGF beta is going to dump a lot of scar tissue on tissue that has been damaged, tissue that is not capable of the capacity to regenerate, the capacity to regenerate. So fibroblasts produce fibers and ground substance of the matrix. They produce fibers and ground substance of the matrix. Macrophages arise from the monocytes. So monocytes are a primordial form, and they're circulating the body. The monocytes are circulating the body, going around the body. Once they detect that something is wrong, they immediately change and they become ready to fight in the form of macrophages. They become macrophages. They phagocytize foreign materials and activate immune system when they sense foreign matter, when they sense the foreign matter antigens. Leukocytes or white blood cells. Leukocytes or white blood cells, they function in immune defense. Examples would be neutrophils would attack bacteria, lymphocytes react against bacteria, toxins, and other foreign agents. Plasma cells. Plasma cells arise from lymphocytes and they synthesize antibodies. The B and the T cells, excellent. These are your lymphocytes. They arise from the plasma cells. Then you have these mast cells that degranulate. These are involved in the allergies, you will see. And they secrete heparin to inhibit clotting and histamine to dilate the blood vessels. Adipocytes. Adipocytes are fat cells clustered in some fibrous tissues. Adipocytes are fat cells clustered in some fibrous tissues where they dominate, called the adipose tissue. So where do we find adipose tissue? We find adipose tissue in the fat tissue. Okay, fibrous tissue. When we examine fibrous connective tissue, we see that components of fibrous connective tissue are the fibers, and we have collagenous fibers, made of collagen, tough and tough, flexible, and stretch resistant. Tough, flexible, and stretch resistant fibers. They are called white fibers due to appearance and their fresh tissue. These are the collagenous fibers. They are found in the tendons, the ligaments, deep layer of the skin, and they are mostly collagen. They are less visible in the matrix of cartilage and bone. Now, when you look at reticular fibers, when you look at reticular fibers, these are thin collagen fibers that are coated with glycoprotein. You tell from the name now, glycoprotein, glycoprotein. This glycoprotein is a hybrid, a mixture, a two-in-one. They form framework of the spleen and the lymph nodes that is part of the basement membrane under the epithelia. There are elastic fibers. These are thinner than the collagenous fibers and made of the protein elastin. The protein elastin is very malleable. It's very malleable. And it allows the stretch and a recoil like a rubber band. Recoil like a rubber band. So these are the ligaments, connective tissue, ligaments on the tendons. What's the difference between a tendon and a ligament? What's the difference? Yeah, but that's halfway. One is attached to the muscle. So it's attached to the muscle and dangling. One is attached to the muscle and the bone. It attaches the muscle to the bone. That's the tendon. Then another one attaches the bone to the bone. That's the ligament. Fibrous connective tissue. Components of fibrous connective tissue. Components of the fibrous connective tissue are ground substance, which are featureless substance surrounding the cells, usually gelatinous or rubbery. They absorb the compressive forces and contains large molecules. Glycosaminoglycans. These are the components of the fibrous connective tissue. Proteoglycans and glycoproteins. There are glycosaminoglycans. These are long polysaccharides composed of amino sugars and uronic acid. They regulate water and electrolyte balance of the tissues. Examples of chondroitin sulfate, heparin, and hyaluronic acid. Proteoglycans are gigantic molecules shaped like bottle brushes. They form gravy-like colloids that hold the tissue together. There's adhesive glycoproteins. These are protein carbohydrate complexes. They bind components of the tissue together. Types of the fibrous connective tissue. You have loose connective tissue and dense connective tissue. Mostly ground substance in the space surrounding the cells. The types of the loose connective tissue are areolar and reticular, and there is the dense connective tissue, mostly fibrous in space surrounding the cells. Types are dense, regular, and dense, irregular tissue. Areolar tissue. There are loosely organized fibers, abundant blood vessels. They possess all 6 cell types and fiber types. They underlie the epithelia in serous membranes between muscles, passageways. They provide passageways for nerves and blood vessels. Now the reticular tissue. The reticular tissue is an example of, you find that one in the spleen. You find that one in the spleen. What is the spleen the graveyard of? You say it, you get extra credit. It's coming out of it. It's coming out of his soul, but it's just not making it out. Well, who said that? Very good. Please give a round of applause. Very nice. Somebody paying attention at 7 p.m. Very nice. Wow. Excellent. So, the spleen is where, they have the American Ninja Warrior. So they test the red blood cell there. There are very tiny passageways that if you make it through them, then you can make it. If you get trapped, you get killed. If you know the names of these passageways, you get extra credit too. What's the name? I'm sorry? What's the name of these tight passageways that's why the red blood cell is biconcave. The bone marrow is in the bone. This is the spleen. I'm sorry? The what? Capillaries are for blood. So the capillaries are they are blood vessels, but these are not blood vessels. These are passageways that are so narrow, and you have your biconcave blood vessel in order to maneuver through them. What are these called? Splenic what? The artery is a blood vessel. This is not a blood vessel. This is like a tunnel for you to get tested for your power. This is called a, nobody's going to know this one.
Oh, sorry.
I'm sorry?
No, I
think she said it.
No, say it. No,
Yeah, she said it. What is it? You looked it up, yeah, you looked it up. Sinusoid. So the sinusoid. Shh, guys, please. The sinusoid is this passageway you gotta make. If you get trapped, you get eaten by a macrophage, but we have different types of macrophages. We have different types of macrophages. You have different types of macrophages. The macrophages of your nervous system are known as microglia. The macrophages of your respiratory system are known as alveolar macrophages. The macrophages of your spleen are known as sinus histocytes. So all of those, so it pays to read the chapter. If you read the chapter, I'm going to be asking these extra credit questions. You say them, you're gonna earn extra credit. So today only one student is going to get the extra credit. So you're going to go home, read the chapter. Read the chapter. Okay. Now what is this? This is collagen. This is connective tissue. So this is densely packed parallel collagen fiber. So that's one of the questions on the lab, not the lecture. On the lab, you will see this photo right here. And there's a question. So now it's like a puzzle. Now you're going to look at this picture and read all around it, just like how it was. You already seen the exam, guys. So it's going to be like that. You're going to see this photo, then you're going to go back and try to tell what is this. Densely packed parallel collagen fibers. These are compressed fibroblast nuclei with elastic tissue that forms wavy sheets in some locations. Tendons attach muscles to the bones, and ligaments hold the bones together. Dense irregular connective tissue. Dense irregular connective tissue. So densely packed, randomly arranged collagen fibers and with few visible cells. They withstand unpredictable stresses, and locations are they are found on the deeper layer of the skin with capsules around the organs. Adipose tissue is synonymous with what word? When you hear adipose, what fat? What color? What color is yellow? Very good. You know, remember the yellow marrow? Yellow marrow. So the adipose tissue is the fat, is the tissue in which adipocytes are dominant. This adipose tissue is what kind of tissue? Epithelial, connective, muscle, or disconnective? All of this been connective. The fiber is connected, the collagen is connective, everything we've been mentioning so far. The reticular tissue in the spleen connected, everything has been connected. The respiratory system macrophages, they are called alveolar macrophage. You know the police in New York called NYPD, police in California called LAPD is the same thing, but it's in different place. It has a different name. So these are all macrophage, but they have different names. So the space between the adipocytes is occupied by areolar tissue, reticular tissue, and blood capillaries. So fat is the body's primary energy reservoir. So the quantity of stored triglyceride and the number of adipocytes are quite stable in a person. So fat is recycled continuously, and new triglycerides are synthesized while old molecules hydrolyzed and released to blood. There are two types in humans. There is white or yellow fat and brown fat. So there is white adipose tissue, what or white fat. That is the most abundant and significant type in the adults. Then it provides thermal insulation. It cushions the organs such as the eyeballs, the kidneys, and it contributes to the body contours. And it's found in female breasts and in the hips. And it secretes hormones that regulate the metabolism. Then there is BOT. There's WAT and BOT. WAT, white adipose tissue. BOT is brown adipose tissue or brown fat. Brown fat is found mainly in the fetuses, in the infants, in the children. So adults have smaller deposits. So it is found mainly in the fetus, in the children. So the color comes from the blood vessels, from the mitochondria and mitochondrial enzymes. Mitochondrial enzymes. It functions as heat generating tissue. Why is brown fat brown? What gives it the color? You know, don't say the fat because the adipose tissue, the yellow one, that's fat also, but it's yellow. Why is this one not yellow? Why is this one, uh, why is this one? Yeah, it's already there. The mitochondria, and mitochondria, and the blood vessels. So that shows you what color is the mitochondria? Brown. So a silly question on the exam would be, which of the following is the color of the mitochondria? Red, green, brown, yellow. Yeah. There's some silly question like that. So yeah. Yeah, so just the color of the mitochondria, because that's the reason why the brown fat is brown. And which of the two fat tissues is important for thermal regulation? There's the brown. That's the heat right here. Heat generating tissue. There you go. That's the yellow. You see that this is the white adipose tissue, or also known as yellow, found in the breast. It is empty looking cells with the margins on the nucleus pressed against the cell membrane, the energy storage insulation, cushioning. Brown fat produces the heat. Cartilage. What structure is cartilage? That's a type of connective tissue. So we did fibrous tissue, that's connective. We did fat tissue, that's connective. The blood, that's connective. We're gonna get to the blood, that's a connective. Cartilage is a stiff connective tissue with flexible, rubbery matrix. Flexible, rubbery matrix. It gives shape to the ear and the tip of the nose and the larynx. So who makes cartilage? It's the chondroblast. Blast meaning they're gonna blast and build up. They're gonna build up cartilage. So cartilage cells, they produce the matrix. There are chondrocytes. These are cartilage cells that are trapped in the lacunae. Lacunae are the cavities. Then you have the perichondrium. The perichondrium is a sheath of dense, irregular connective tissue that surrounds elastic and most hyaline cartilage. Surrounds elastic and most of the hyaline cartilage. It contains a reserve population of chondroblasts that contribute to the cartilage growth throughout the life. General features of the cartilage. So there are no blood capillaries. There's the so cartilage is avascular, has no blood capillaries. Diffusion brings nutrients and removes the waste, and it heals slowly. The matrix is rich in glycosaminoglycans and contains collagen fibers. The types of cartilage vary with fiber composition. So there is hyaline cartilage, fibrocartilage, and elastic cartilage. So what kind of cartilage are we dealing with? You can tell elastic cartilage is what kind of cartilage? Is very malleable. So elastic tissue is very malleable. Now, hairline tissue is like glass. Hairline tissue is like glass. So there is clear, glassy appearance because of the fineness of the collagen fibers. Because of the fineness of the collagen fibers. It eases the joint movement. It holds the airway open and moves the vocal cords. And that's important for the growth of the juvenile long bones. Which of the cartilage is glassy? Is it hyaline, fibro, or elastic? Hairline. That's glassy, clear glassy appearance because of the fineness of the collagen fibers. It eases the joint movement. It holds the airway open and moves the vocal cords. And it's important for the growth of the juvenile long bones. Locations are the articular cartilage, costal cartilage, trachea, larynx, and fetal skeleton. That's another question on the lab exam, not the lecture, the lab exam. This photo right here. So that shows you what kind of cartilage is that. That's elastic cartilage. That's the cartilage containing abundance of elastic fibers covered with perichondrium, and it provides flexible elastic support, and locations are external ear and epiglottis. Fibrocartilage. Now that's the one that is found. So you should be able to tell where I can find hairline cartilage. Where can I find hairline cartilage? Trachea. Where can I find elastic cartilage? From the name, elastic cartilage. That's my ear. Very good. And where do I find fibrocartilage? That's in the vertebral column. That's the cartilage containing large, coarse bundles of collagen fibers, and it resists compression and absorbs shock. Locations are the pubic symphysis, the menisci, and the intervertebral disc. Who's the last type of connective tissue? That's your bone. So bone is an osseous tissue, is calcified with a lot of calcium salts. Is a calcified connective tissue that composes the skeleton. There are two forms of osseous tissue. Two forms of osseous tissue: spongy bone and compact dense bone. Spongy bone is a delicate, is made up of delicate slivers and plates that give it a spongy appearance. It is found in the heads of long bones and in the middle of the flat bones, such as the sternum. It is made up of compact, dense bone with denser calcified tissue with no visible spaces. It forms external surfaces of all the bones. So what are the features of compact bone? It is arranged in cylinders known as osteons. It has a central osteonic canal with passage running longitudinally along the bone shaft, containing blood vessels and nerves. So now there is the concentric lamella. The concentric lamella are a ring-like layers of bones surrounding the central canal, and you have the osteon is a form of a fluid. This fluid that is secreted that would become hard and become bone. So the central canal is going to be secreted in the central canal surrounding the lamella, and osteocytes. Osteocytes, these are the mature bone cells within the cavities, the lacunae. Canaliculi. The canaliculi are a delicate canal radiating from each lacuna to its neighbors, allowing the osteocytes to contact one another. The periosteum is a tough, fibrous connective tissue covering the whole bone. So covering the skull is the periosteum. So you will see now, you see, where do I find compact bone? That is found in the femur. You see, it's a cylinder that surrounds the central osteonic canal that runs longitudinally through the shaft of the long bone. The blood vessels and the nerves travel through the central canal. The bone matrix is deposited in the concentric lamella. Now, don't forget about your blood. Blood is an example of a connective tissue. So, blood is a fluid connective tissue that travels through the tubular blood vessels, that travel through the tubular blood vessels. They transport cells and dissolved matter from place to place. They transport cells and dissolved matter from place to place. They contain formed elements suspended in a liquid ground substance known as the plasma, the blood plasma. So you know when you centrifuge the blood, when we do the blood chapter, you will see that taking an example of blood, centrifuging that. When you centrifuge the blood, what do you get? You get three layers. The heaviest layer is going to be at the top or the bottom. And it's going to sink first or last? First. Very good. Why first? Excellent. And the middle is going to be what layer? The buffy coat, and that's going to be those white blood cells and platelets. Because why do we not have a lot of them? Because they are made on demand. I don't get cut all the time, so I don't need platelet all the time. I need platelet sometimes when I'm bleeding. I don't need white blood cells all the time. I only need white blood cells when I have a microbial infection, parasitic infection, bacterial infection, viral infection. blood smear. So the erythrocytes appear as pale pink disc with light centers. Leukocytes are slightly larger and have various shaped nuclei. And platelets are small cell fragments with no, they are fragments of what? They are fragments. Fragments from what? You fragment from what? Bone marrow. So the platelets are fragments from the bone marrow. Now, lastly, you see how they put them together. Nervous and muscular tissue. They come together as the same, they come together as a one form of tissue known as the excitable tissue. Okay, so what is excitability? Excitability is the ability to respond to stimuli by changing the membrane voltage. So how do we change the membrane voltage when electrolytes flow in, or when there's an influx of electrolyte or an efflux of an electrolyte changing the membrane potential? So it is developed to highest degree in nervous and muscular tissues. So they are called excitable tissues. So what are excitable tissues? Tissues that respond to stimuli by changing their membrane potential. So excitation founded in the charge difference, known as the voltage across the membrane, called the membrane potential, In nerve cells, changes in voltage result in rapid transmission of signals to other cells. So in muscle cells, changes in voltage result in contraction and shortening of the cell. Now that's the difference. You got to know that one. When you compare a nerve cell who is excited, obviously both of them are excitable. This is known. We know that both of them, nerve cell, muscle cell, are both types of excitable tissue. So nerve cell and muscle cell are both examples of excitable tissue. Both get excitation. Both are excitable tissue. But when the nerve cell gets excited, it transmits information. But when a muscle cell gets excited, what happens to it? It contracts, gets shorter. It goes from here to here. You get it? So, I'm sorry? So a membrane potential change. So you're going to see the opening of the channels, and you're going to see that there's flow, influx. When you do that video, that 5-page video, when you listen to me explaining, you will see that we're going to have the influx and the efflux, and that's going to change the voltage. That's going to change the voltage, and that's how we mediate through the action potential. We're going to get to the action potential. That's how we're going to be able to mediate the information. So, I'm sorry? Yeah, through the neurons, and the neurons, they're gonna get excited. Then they're going to send a neurotransmitter that's going to go and bind to receptors on the muscle cell, causing the muscle cell to contract. We're gonna get to that. Nerve tissue is specialized for communication by electrical and chemical signals. They consist of neurons, nerve cells that detect the stimuli, respond quickly, and transmit coded information rapidly to other cells. There's neuroglia. Neuroglia are glial cells, not microglia. Microglia, that's the macrophage. This is neuroglia. These neuroglia, these are the housekeepers of the nervous system. These are like the nerve cell. These are not nerve cells. They are supporting to the nerve cell. So in the police precinct, you don't only find police officers. You find HR there. You find secretaries. So they support the police. So the neuroglia, these are the ones that give you brain cancer. God forbid. You know, the nerve cells, they never divide. Nerve cells cannot enter the cell division cycle. Nerve cells can never undergo mitosis. Nerve cells are in a state of terminal senescence. They are quiescent cells. But the other supportive cells, like the ependymal cells, as you will see, who make the cerebrospinal fluid, like the Schwann cells and the dendritic cells, the oligodendrocytes. These are the cells that make the myelin sheath around the axon. They are the ones who have the capacity to enter the cell division cycle. So they are the ones who make the brain cancer, not the neuron itself, but them. Parts of a neuron. The cell body houses the nucleus and the other organelles, and it controls protein synthesis. Dendrites are short branched processes that receive signals from other cells and transmit messages to the cell membrane. And the axon is the nerve fiber that sends outgoing signals to other cells, and it could be more than a meter long. We're gonna have our break when we finish the lecture. We're gonna have our break, and then we're gonna do the lab. And as soon as you finish the lab, you learn everything, then you can go. I don't know what do they want us to do? Stay till the 8. We saw the slide, right? You saw the slide. You're gonna see the slide. You saw the slide, and we're gonna see it again. And then what else can we do? I explained, right? We recorded that. So let's do the lecture, and then we take a break, then we go to the lab. We're gonna have a picnic. We're gonna sit, sing, wade in the water. So we can't do that. We finish the lab. We go home. That's it. Okay, neurons and glial cells. Neurons and glial cells. So you have the cell body, the dendrites, and this is the axon. So you must know that the axon is how we transmit the information. So the cell body is where we process, the information, and the axon is where we send this information. Okay. So muscular tissue, is specialized to contract when stimulated, exerting a physical force. So you see, they contract when they are stimulated. And they are an important source of heat. That's why you shiver with your muscles. You don't shiver with your skin, you shiver with your muscles. And shivering is going to do what? Increase heat or decrease heat? Increase heat. Cardiac and smooth muscle. They are made of muscle fibers that are long, thin cells, most skeletal muscles attached to the bone, and they contain multiple nuclei adjacent to the plasma membrane, and they must have striations. You know, we have different types of muscle cells. We have cardiac muscle, muscle of the heart. That's a muscle too. Don't think muscle is your bicep only in the gym. No, that's a skeletal muscle. There's also cardiac muscle. In addition, then you have smooth muscle, the blood vessel. That's a type of a muscle as well. Okay. Okay, let's have a 10-minute break, then come back. We finish the lecture, then you have your big break, then you have our lab. Okay? Who said that? Okay, I will do my job. Okay, you just take your break. Take your break, and then we 10 minutes, and then we come back immediately to take the test.
Come back for the best
How long were you doing it?
Hello, baby.
I like chocolate.
You're like,
Yeah.
no,
I'm kidding. I want good karma, that's all. Good karma.
many blessings are waiting.
Thank you, I need it. I'm clear. I hate this break and then we'll take a real break. No. We didn't even need the 10 minutes, we could just wait, and then if you want to take a real break, we. So this 10 minutes be a half hour. So good. Yeah, because usually when it's recorded, it's like the oh, like the game. It was a, I don't know if you guys seen it, but it was viral for us. And we're probably like, this girl had an interrogation with the cop, and I guess they're like looking for her boyfriend. And she was like, I don't know, I haven't spoken to him since he told me he was going to connect over to Connecticut. And she was dead ass like a dick. The cop couldn't even stop laughing at her. On a train. It is the same driver, however, the train you don't get caught in traffic. I missed the quiz, that's why I stopped driving. I missed the quiz in the morning and I couldn't make it up and that zero had me up for the entire thing. That's what it lets you make up an exam, but quiz is, yeah. So I got here, if I would have made it and somebody should have taken the quiz, I would have allowed to take it because everybody was done. He didn't have to take it, I had a zero, even though he dropped it then later on, but that's not gonna be the case with everything. So that's just, I like, for me. Yeah, a lot of them know what you're supposed to do. No problem. But then, to get those progressive, like today, my stuff is not, so we can help with it, so we got to do today. It'll be like, I don't say that again. I mean, yeah, I had a class on Friday. I had to do it. The guys in 9 to 12 about around.
Okay, guys. All right. So we were trying to manage which lab we're gonna get to. There's another class also. Okay. So we have okay, we just have this to finish, then we gotta go to lab. We just have all right, 30 slides, then we'll go to lab. Okay? No. Okay. All right, 30 more slides so we can go to lab. Let's go, guys. Okay. Okay. Limited. So we know we have 3 types of muscle. We have cardiac muscle that is limited to the heart wall. There's cardiac myocytes. These are short and branched with one centrally located nucleus, intercalated disc joint cardiomyocytes end to end. And there are striated and involuntary. They are striated and involuntary. Smooth muscles, they are made of short fusiform shaped cells. Cells have one central nucleus with no striations, and they have an involuntary function. Okay. Okay. You know now, cellular junctions, these are like staples. They connect between 2 cells. So most cells are anchored to each other or their matrix. So the cells communicate with each other, resist mechanical stress, and control what moves through the gaps between them. So you know the cells, we have to know that cells must communicate. So sometimes they have open gaps, they have tunnels to pass electrolytes and information through. Sometimes we have caps, cell adhesion proteins that hook and anchor the cells together. And there are tight junctions. You see, these are the zipper-like. They hold the cells in place. And then there are the desmosomes. Desmosomes are patches that hold the cells together, like a clothing snap. And you know the gap junctions, these are the communicating junctions. That's an excellent depiction. So you see this figure right here, you study that for your lab exam after the lecture exam. You see those are the tight junction, these are the zipper-like, they hold the cells together, and these are the desmosomes, and these are the holes, so tunnels, they allow for the passage. These are the connexins. They allow for the passage through the electrolytes. There is the intracellular space, plasma membrane, and cell adhesion proteins. These are cell adhesion proteins, plaques, and intermediate filaments of the cytoskeleton. These are proteins, that's the connexin, and this is the pore through which the material is going to pass. Okay, so glands, these are cell or organs that secrete substances for use elsewhere in the body or releases them for elimination from the body. So they excrete waste and they secrete product that is useful to the body, like a hormone or some form of enzyme or sweat. Exocrine glands, they maintain their contact with the surface of the epithelium by way of the duct. And there are endocrine glands, they have no duct, but do have many blood capillaries, and they secrete hormones directly into the blood. Okay, these are the types of the different glands. That figure is also on the exam. So here you have to examine that an endocrine gland is near a blood vessel. See the blood vessels? Going to empty the content into the blood vessel. And this is an exocrine gland, is going to empty its content into a duct, into a duct. So an endocrine gland in the blood, exocrine gland in a duct. You see where my mouse is? My mouse is exocrine duct, endocrine blood vessel. Very important to know the difference between an endocrine and an exocrine gland. The pancreas, is it an endocrine or an exocrine gland? Endocrine and exocrine.
Oh, yes.
It secretes insulin into the blood into your blood, insulin into your blood. It secretes glucagon into your blood, but it also secretes those digestive enzymes, pancreatic lipase to break down lipids. That is also from the pancreas. You see, that's the general structure of the endocrine and exocrine gland. Okay, that's the capsule. Okay. The tubuloacinar. And that shows you an example of these are the different types of the exocrine glands. This is a compound tubuloacinar gland that is found in the pancreas. This is a compound acinar gland, and that's a simple coiled tubular gland. Okay. Okay. These are three modes of exocrine secretion. You have eccrine, that secretes to the extracellular space, and then there are the apocrine that secrete in the form of bubbles and vesicles, and you have the holocrine that internalize their content to the inside. And we have different types of membranes. We talked about those, the mucosal membrane and the cutaneous membranes. And you see right below the base, you see the basement membrane? We talked about that. The basement membrane, right below it is the blood vessels. You have serous membranes. This is serosa. You find that where? You find that in your stomach, in the cavity, in the pleura, in the pericardium, and the peritoneum. So we have hyperplasia, hypertrophy, and neoplasia. We talked about the metaplasia. If you put so much nicotine to your esophagus, it's going to change into a tougher tissue. That's metaplasia. But then if you change to a cancer, that's anaplasia. So metaplasia is when you change to adapt, to accommodate, to accommodate to the environment. Okay. So stem cells, we talked about them being progenitor cells. These are progenitor cells that have the capacity. We see them with blood vessels. They would become colony forming units. Upon receiving the proper cytokine, they would differentiate into the proper lineage. Do we want red blood cell? Then we would send a particular cytokine to lead the stem cell to become red blood cell. We want a white blood cell, we send a cytokine to make the stem cell become white blood cell. Which white blood cell? Eosinophil, basophil, neutrophil. That is all based on what type of tissue. There is regeneration and fibrosis. So regeneration is when the cell divide. After the cell dies, new cells come in its place, actual cells. But if the tissue is non-regenerative, can never enter the cell cycle, then we dump fiber there. We dump scar tissue. That's known as healing by fibrosis. So that's the healing with scar tissue, and we talked about the necrosis and apoptosis. Okay, let's take a break, and we come back to lab. Let me take the attendance first. Depending on how many students we have, we're gonna go to the appropriate lab.